Hello, welcome to EPG Pathshala. I am Dr. Mariam Sada, Associate Professor in the Department of Biosciences, Jamia Millia Islamia, New Delhi. Today I am going to teach you about the nano biocatalysis. In today's lecture, we will talk about nanomaterials and the approaches to synthesize these materials. We will also discuss about the application of nanomaterials as immobilization matrix. The different methods to conjugate a protein or enzyme to these nanomaterials will also be discussed in this lecture. So, what are nanomaterials? Nanomaterials are one of the main products of nanotechnologies as nanoscale particles, tubes, rods or fibers. Nanoparticles are normally defined as being smaller than 100 nanometer in at least one dimension. Natural nanomaterials are volcanic dust, incidental include diesel exhaust, carbon nanotubes, and the metal nanoparticles are examples of engineered nanomaterials. So, we can say the size of the nanoparticles ranges from 1 to 100 nanometer. We will discuss about the properties of nanomaterials. Nanomaterials are stronger and more ductile materials and they are very active components. They have high strength, toughness and ductility. Their physical and chemical properties are very different as compared to bulk materials. They are easy to prepare to form a colloidal solution. Now we will see how we can synthesize these nanomaterials. There are three methods for synthesis of nanomaterials. They are classified as physical, chemical and biological methods. Each have their own advantage and disadvantage. Biological methods of synthesis are preferred over other methods when nanomaterials are applied in biology and medicine. There are two general approaches to synthesize the nanomaterials, bottom-up approach and the top-down approach. The top-down method includes physical methods and the bottom-up method includes chemical and biological methods. This slide shows the comparative study of the different types of methods of synthesis of nanomaterials. As biological methods are cost effective, mild reaction conditions are required, no toxic byproducts and considered as eco-friendly. Here a flow chart of the synthesis of nanomaterials using biological approach has been shown. The metal nanoparticles can be synthesized using plants or plant extract, microorganisms or biomolecules. Mainly the biomolecules are responsible for the reduction of metal ion into the nano form. Here one can see the different types of nanomaterials like nanoparticles, nanocomposites, nanopores, nanofibers, nanotubes and the nanosheaves. These nanomaterials are used in biology, biotechnology and medicine. In this lecture, we will discuss about the applications of nanomaterials in biocatalysis. Nanobiocatalysis is dealt for enzymes which are incorporated into nano-structured materials. Nanobiocatalysis is dealt for enzymes which are incorporated into nano-structured materials. Now we will talk about the nanomaterials as immobilization matrices. Nanomaterials are ideal matrix for protein or enzyme immobilization because they possess large surface to volume ratio. Smaller particle size reduces the size of the bioreactor. When dispersed in aqueous solution, they show Brownian movement which increases the rate of reaction. Various strategies are there to linking an enzyme to the nanomaterials. They can be classified as electrostatic adsorption, covalent attachment, conjugation to a specific affinity for a protein, direct conjugation to the nanomaterial surfaces. In electrostatic adsorption, enzyme is attached to the nanomaterial by physical method. The non-covalent interactions play important role in binding the enzyme to the nanoparticle. Surface of the nanomaterial is modified in covalent attachment so that the enzyme can bind to the nanomaterial. There are a number of chemical reagents available which are used to modify the surface of the nanoparticles. Another way by which enzyme can be linked to the nanomaterial is by affinity linking. An affinity ligand which is specific to the protein of interest is attached to the surface of nanomaterial and it is specially bind to that protein only. Some nanomaterials like silver and gold nanoparticles interact with the thiol group of protein without the use of any linker. Now, this slide shows the advantages and disadvantages of using nanoparticles for enzyme immobilization. Of course, we have lot of advantages of nanomaterials for using an immobilization matrix. Some disadvantages are also there as they are cost effective. Talk about the application of nano immobilized enzyme systems. Nanostructure supports are used for the immobilization of enzymes in order to improve the bioreactor performance. The enzyme immobilized on nanomaterials can be used for the synthesis of sugar esters or for biodiesel production. They are also used as biosensors, biofuel, etc. Biofuel cells are a type of fuel cells that employs biological components as the catalyst for the oxidation of fuel. Application of nano immobilized enzyme as a biosensor. The biosensor is usually composed of three parts: a sensitive biological element such as a cell receptor, enzyme and antibody, a transducer or a detector associated with electronic and the signal process. Based on the transducer type, the biosensor can be classified into an optical, colorimetric, piezoelectric, conductometric, potentiometric and amperometric biosensor. This slide shows some of the enzymes immobilized on different kinds of nanomaterials and their applications. To summarize today's lecture, we can say that nanomaterials, owing to have large surface area, are the preferred matrices for protein or enzyme immobilization. Another important property of nanomaterials is that their surface can be easily modified, so large amount of molecules can be attached to them. The protein can be attached either by physical adsorption or by covalent methods. Use of nanomaterials reduces the size of bioreactor, which is required in biotechnology industries. So, to summarize today's lecture, we have learnt about nanotechnology, nanoparticles and nanobiocatalysis. We have also discussed about the different approaches to synthesize nanoparticles and their application as a matrix for nanobiocatalysis. Thank you. You can also search this material at UGC Pathshala program.